Hello everyone. Since we studied the biotechnology application of biotechnology, I wanted to look at your book, which is page 160. Here you have like a technique. He's a scientist. It's a scientist. He's holding something on his hand. It's glowing. We want to talk about this glowing band. Actually, as you see here, these are called what? A fragment of DNA that have been treated with a special chemical, and this special chemical glows under ultraviolet light, allowing scientists to see the DNA. Why the scientists they are using this? What is this lab technique? Actually, this one is called electrophoresis. What is the electrophoresis? Actually, here. As you see in the graph here, here they put it's a technique for analyzing DNA. Electrophoresis actually again is a technique. I'm going to write everything about it. I want you to, I'll ask it, I'll show you everything about these notes. Okay, is a technique for analyzing DNA. So electrophoresis applies an electric current. Actually, here it applies on this plate here an electric current, okay, that travels from one end of a depression filled with a gel material. Yani this one here, it has a gel material, okay. So an electric current can pass through it. A DNA sample that have been cut by digestive enzyme. Yani before you applying this one, you bring a DNA sample, and the DNA sample, it has been cut by a digestive enzyme. They will be placed in these wells, yani here. Yani one will be here, the other one will be here, other sample, here another sample, okay. Then an electric current can pass through it, okay. So the current moves toward the opposite end of the gel. Fragments of the DNA also move while it's moving from this side to this side, for example, from this side to this side, okay. So the current moves toward the opposite end of the gel. A fragment of the DNA also move. Smaller fragments move farther through the gel than larger fragments. After a set amount of time, the electric current is turned off. Actually, the stain is used here in electrophoresis, which is ethidium bromide, which is used to stain the DNA to make it visible under what? Under the ultraviolet light, okay. So electrophoresis can be used to help isolate particular genes to study or to create a DNA fingerprint that can be used to identify individuals, such as using in forensic evidence, okay. I'm going to show you now a video of electrophoresis to understand it. Maybe if I'm talking about it in this way, you won't understand. So if you look at this video, it will help you to understand this lab technique. So listen to this video carefully to understand exactly what is electrophoresis.
I kind of thought that when two organisms had a baby, the baby was just this blend of the two. Yeah, that's a misconception. But I really saw genetics in action with my guppies. Guppies are very easy freshwater fish to keep in an aquarium. But they have two things that I think are especially cool. They have live birth, which means there are no eggs, like many other fish. And second, they have a lot of babies. They also eat their babies, but I don't think that's especially cool. So as you can see, that is not part of my cool fact list. Anyway, when my surviving baby guppies grew up, they would have all kinds of cool traits. These traits were carried by their DNA, their genetic material, which is found in their body cells. But sometimes I would forget which mother was the mother of the baby guppy, because there were several mother fish in the tank, and I wanted to keep track of inheritance in my guppy notebook. So what would have been very cool to have at that time? Some biotechnology. Biotechnology is the merge of biology and tech, and it's constantly changing. It includes topics such as PCR, cloning, and genetic engineering. It's also an awesome field. We're going to talk about one of the biotechnologies that could have, well, potentially, helped me determine the genetic relationships of my guppies. If, as a young kid I had access to it, although it's becoming way more common in classrooms now. And that biotechnology is gel electrophoresis. Gel electrophoresis can be used to separate.
I want to stop here. I want to show you, I was talking about the wells. These are the wells where you put the DNA sample, and how when you start to allow the electric current to pass through it, how these are fragments of DNA are here. I want you to listen carefully to see. I'm going to repeat this part.
As a young kid, I had access to it, although it's becoming way more common in classrooms now. And that biotechnology is gel electrophoresis. Gel electrophoresis can be used to separate molecules based on how big they are, their size, and it's especially useful with DNA. Let's look at DNA real quick. So here is a guppy cell, here's the nucleus in the guppy cell, here's the DNA in the nucleus of the guppy cell, and if you were to zoom into the DNA, here is a nucleotide, which is a building block of DNA. See those phosphates in the nucleotides? They're a bit negative. Well, they contribute a negative charge anyway to the DNA. So if we look at this whole DNA here, it gives that DNA a negative charge. That's a big deal because gel electrophoresis, which again separates molecules based on size, relies on the fact that DNA molecules have a negative charge. Okay, here's a gel electrophoresis machine. The point of the machine is to be able to have an electrical charge running through a gel. So here's the gel, typically made of agarose. Agarose is a polysaccharide polymer, which, if you remember from our biomolecule video, polysaccharides are carbohydrates. Yeah, usually agarose comes from seaweed. The agarose gel itself lets the DNA molecules travel within it. One end of the gel has these holes called wells. The wells are where the DNA is placed into. The area of the gel where the wells are is negatively charged, and the area of the gel here is positively charged. So guess where the DNA will travel towards? Well, since it's negatively charged, it's going to travel to the positive side. So typically when you're analyzing DNA in electrophoresis, you use these restriction enzymes to cut the DNA up into tiny pieces. Restriction enzymes have the ability to cut up DNA in very specific areas, often related to the specific DNA bases, making restriction enzymes very useful in biotechnology. So if I had baby guppy DNA and adult mother guppy DNA, and I want to compare them, then I would want to use the same types of restriction enzymes in both DNA samples. If I use the same type of restriction enzyme, it should be cutting the DNA at the same identification points in the DNA samples. However, unless the mother and baby guppy are clones, and they're not, those pieces that result after the restriction enzyme is done with them may be differently sized because the DNA of the baby and mother guppy had some differences in the sequence of their DNA bases. So the DNA samples both are cut up into multiple pieces by the same type of restriction enzyme, and then those samples are loaded into the gel, sample 1 and sample 2. If we turn on the machine and let the DNA run through the gel, the DNA moves towards the positive side. But some pieces of that cut up DNA will move faster or slower than other pieces. Longer DNA pieces tend to have a higher molecular weight. They take more time to make it across the gel when you compare it to shorter DNA pieces which move at a faster rate. So what you end up with is that these DNA fragments spread out, with the longer pieces closer to the wells and the shorter pieces closer to this opposite side of the gel. These are called DNA bands, but to see them, you usually need to stain the gel itself and view it under a UV light. Now let's compare the DNA bands in this hypothetical, simplified guppy situation. The bands aren't going to be identical because these fish are not clones, but I can compare how similar the bands are and compare that to other mother guppy samples to look for relationships. Let's say that we have 3 mother guppy samples to view, and these are the only possible mothers from the fish tank. Which one appears to be the most related to the offspring in this case, and has a high likelihood of being the mother? Well, this one. But we can't be 100% sure with this. It would be helpful for me to know the father guppy sample too, because this will give me more insight. But if these are the only fish in the tank, it's a very high likelihood with this case. Also, you can use something called a DNA ladder. You can buy them from various science material distributors, but a DNA ladder is not what it sounds like. It's basically a sample that has known fragment sizes. So if you run it in the electrophoresis machine, you already know the fragment lengths. Let's say this DNA ladder only had 3 bands, which is not usually the case. Since it's a DNA ladder, the base pair lengths are known. They are 500 base pairs, 1,000 base pairs, and 1,500 base pairs. Think for a minute. Where would they fit in?
I don't care about this, but I want you to understand what is the electrophoresis. Only the definition. This one is not very. It's not required from you, but you can listen for it for your information.
It would look like this. You can use this now as a reference to give estimates of how large the other fragments are when they're run alongside it. And if you want to be closer to the value, you can use a semi-log graph, something to look up. So why do we care about gel electrophoresis? It's not likely I'll be actually using this with my guppies anyway, right? Well, perhaps. But gel electrophoresis is often a step used in determining relatedness with different species, which helps scientists better classify organisms. It's also used as part of DNA fingerprinting. DNA fingerprinting is a way that one can identify someone's DNA, which can be very helpful if you're trying to solve a mystery involving a crime scene. If you have a DNA sample from a crime scene, you can go through the steps of gel electrophoresis to compare it to the suspect DNA to see the likelihood of a match. In fact, you can take the results from gel electrophoresis and isolate genes of interest by something called southern blotting. Definitely something to look up if you're curious. Gel electrophoresis is one of many awesome tools in biotechnology. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So this is all about the electrophoresis. I want you now to look at this one here, just to sorry for this. I want you to copy this just explanation what is the electrophoresis. It's talking about the figure in your book, a photograph in your book actually, but here you can copy this information to understand what the electrophoresis. If you have any question about electrophoresis, you can send me your questions. Thank you for listening.